Good morning everyone. Myself Roshan Katare. Today we as a team from roll number 81 to 90 going to present seminar on abnormal hemoglobin. This is how we going to proceed in seminar. So our friend Prathamesh Soni is gonna talk about normal structure of hemoglobin.
To understand about the abnormalities in the hemoglobin, let us understand the normal structure of the hemoglobin so that we can understand what are the abnormalities into the structure or any kind of mutations occurs. So normal hemoglobin is a conjugate protein, globin that apoprotein and in the non-proteinous part of the prostatic group. So as we can see into the figure, which you can see into the brown color, two chains that are the beta globin chains and the pink color chains are the alpha globin chains, and in that you can see a Saturn-like projection or the red color part which is of the iron or the which helps into the binding of the oxygen. And the structure of the globin consists of 4 polypeptide chains of 2 alphas and 2 betas as we see into the figure. Then the structure of the characteristics red color of the hemoglobin is ultimately blood, which is due to the heme present into the hemoglobin, and the heme consists of porphyrin molecules, namely porphyrin 9 with iron at its center. As we can see, this red color 4 structures in the center of each of the globin chain, that is iron. Now we are seeing the normal hemoglobin. Let us understand the abnormalities. Abnormality hemoglobin are the result of the mutation of the gene that codes the alpha and the beta gene. So as we can see into the figures, the first figure shows the normal lumen of the blood stream in which the biconcave cells RBCs are there. So and in the second part we can see there are the RBCs but which are sickle shaped. This is due to the mutation into the 6th position of the normal hemoglobin, where usually glutamate is present, but due to the mutation instead of glutamate there is the valine. And about the sickle cell anemia you will be further understanding in detail after a bit. Now the classification of the abnormal hemoglobin will be given by Rudresh Bhuya.
Good morning everyone. Before proceeding to the classification of abnormal hemoglobin, first we must understand how abnormal hemoglobin is formed. For that purpose, as you all know, the hemoglobin is made up of alpha, beta, gamma and delta chains, which are made up of amino acids. The gene mutation in this gene results in formation of abnormal hemoglobin, and which are further classified on the basis of 2 defects, which are qualitative one and quantitative one. The qualitative one includes sickle hemoglobin, which causes sickle cell disease, hemoglobin C and hemoglobin D, which causes mild anemia, and hemoglobin E, which causes anemia. And quantitative one includes thalassemia. Thalassemia results due to reduced production of alpha and beta chains of the globin portion of the hemoglobin. And the point here to be noted is that this can be identified on the basis of electrophoresis pattern. For better understanding, let's have a state play.
Now we will present a small script to demonstrate the abnormal hemoglobin.
राम या बसा
राम केशवराव.
हा नक्की तुमचा मुलगा पुष्पराज असणार.
हो, हा आमचा मुलगा पुष्पराज. मागच्या वर्षी इंजिनिअरिंग पूर्ण झाली साताऱ्यामधून. आता अमरावतीला जॉब म्हणून असतो. आणि तुमची मुलगी
शिवल्ली.
बरं मुलगी सुटली की लावून टाकू लग्न.
पण बाबा मला वाटतं लग्न आधी ब्लड टेस्ट करायला पाहिजे.
काही गरज नाही ब्लड टेस्टची.
अहो ते म्हणतात ना काही गरज नाही, कशाला काय.
तर ठरलं पुढच्या आठवड्यात लग्न लावून टाकू.
तर अशाच प्रकारे पुष्पा आणि श्रीवल्लीचं लग्न ठरलं. 8 दिवसात त्यांचं लग्न झालं आणि 9च महिन्यात त्यांच्या घरी एक गोंडस बाळ जन्मायला आलं. तो मुलगा मोठा झाला, खेळू बागडू लागला आणि असंच एक दिवशी मैदानात खेळत असताना
काय झालं तुला? डॉक्टर डॉक्टर अचानक खेळता खेळता पडून गेला.
सर पेशंटची रिपोर्ट.
त्याला सिकलसेल ॲनिमिया झालेला आहे. हा एक ब्लड रिलेटेड रोग आहे ज्याच्यामुळे ऑक्सिजनची कमतरतामुळे लाल रक्तपेशी सिकल म्हणजे हसावी वृत्ती होतात. हा एक नॉन इन्फेक्शियस रोग आहे आणि हा आता मुलांमध्ये आहे.
पण असं तर आम्हाला काहीच नाही.
हे आवश्यक नाही की याचे लक्षण तुमच्यामध्ये दिसावेच. नाही याला बोलणार ट्रान्सप्लांट करावं लागेल.
त्याला किती खर्च येईल?
साधारण 4-5 लाख रुपये खर्च येतो किंवा तुम्ही दर महिना ब्लड ट्रान्सफ्युजन पण करू शकता.
पहा मी म्हणत होते आपण ब्लड ट्रान्सफ्युजन आपण लग्नाआधी मी म्हणत होते आपण ब्लड टेस्ट करूया, पण तुमचे बाबा ते म्हणाले नका करू. पहा आता बघा काय झालं माझ्या बाळाला ते.
हो, आता मलाही पश्चाताप होतोय. आपण लग्नाआधी ब्लड टेस्ट करायला पाहिजे होती. फक्त कुंडली जुळवणं हे महत्वाचं नाही, पण ब्लड टेस्ट केली असती तर आज माझा मुलगा असा झोपलेला नसता इथे तुमच्यासमोर. तर आता आमच्या वेळेला तरी आम्ही रक्त तपासणी केली नव्हती, पण आता तुमच्या वेळेला असं काही होऊ नये किंवा तुमच्या बाळासोबत असं काही होऊ नये म्हणून लग्नाआधी कुंडली जुळवा, पण त्याच्यासोबतच रक्त तपासणी सुद्धा करून घ्या, जेणेकरून पुढे तुमच्या मुलाला काही आजार वगैरे होणार नाही. धन्यवाद.
Good morning everyone. So now I will start what is sickle cell anemia. So basically sickle cell anemia is a genetic disorder which is caused by abnormal hemoglobin, that is sickle hemoglobin HBS. In this the erythrocytes of the patient get sickle shape due to low oxygen concentration. In sickle cell anemia there is mutation of the HBV gene of the beta globin chain. In normal hemoglobin structure, there are 2 alpha chains and the 2 beta chains. But in sickle cell anemia, there are 2 normal alpha chains and 2 abnormal beta chains. In this that is due to change in the single amino acid. So basically, here we can see there is change in a single nucleotide, which is adenine takes place of the thymine in the gene. And Due to this error, there is change in the altered codon which is formed, that is GUG is formed in the place of GHG, and there is formation of valine in the place of glutamic acid. So basically here we can see this condition takes place if there is inheritance of both the mutated genes from the parents, then it is homozygous condition, it is called as sickle cell disease. And if there is only inheritance of only one mutated gene from the parent and the other is a normal gene, it is the heterozygous condition and it is called the sickle cell carrier. In the case of sickle cell carriers, their condition is normal, they do not show any symptoms. In the sickle cell traits, they show resistance to malaria. As malarial parasites have an obligatory part of their life cycle, they show in the RBCs, but as the lifespan of sickle cell RBCs are very short span, so they cannot complete their life cycle. So here we can see has the malarial parasite entered in a normal RBC and it is causing RBC malaria. But in the sickle cell, they enter but they cannot complete their life cycle and thus malaria is not caused. The abnormalities related with sickle cell anemia, first is lifelong hemolytic anemia, second is tissue damage and pain, third increased susceptibility to infection and premature death. Now the mechanism of sickling and sickle anemia. Glutamate is a polar amino acid and it is replaced by valine, which is a non-polar amino acid. So basically this causes formation. This causes formation of the sticky patches on the outer surface of the beta globin chain. Due to this formation of the sticky patches, this is present on deoxy-HBS as well as on the oxy-HBS, but they are absent on adult hemoglobin. So there are some receptor sites present on the deoxy-HBS. The sticky patches of this deoxy-HBS, they get attached to the receptor sites of the deoxy-H and this continues till there is formation of long polymer chains of HBS. And this long polymer chains, they precipitate and they distort the sickle-shaped RBCs. Now in the case of oxy-HBS, there are presence of sticky patches, but there is the complementary sites they get attached. So basically, this mechanism is only seen due to polymerization of deoxy-HBS. Now second, there is diagnosis of sickle cell anemia. Sickling test, in this there is a blood smear is prepared by adding agents such as sodium metabisulfite, and in the second process there is electrophoresis. Electrophoresis at the alkaline pH shows a slower band of HBS than HBA, as glutamic acid has a negative charge and there is absence of glutamic acid and sickle hemoglobin. Therefore it is lower than adult hemoglobin, as we can see here in this. And this management of sickle cell anemia, administration of sodium cyanide inhibits sickling of erythrocytes, and in patients with severe anemia, repeated blood transfusion is required. Now, further thalassemia will be continued by Rahul.
So, good morning everyone. Let's discuss thalassemia in detail. Thalassemia is not a single disease, it is a group of disorders that are hemolytic disorders characterized by impairment or imbalance in the synthesis of globin genes. There are basically two types of thalassemia, alpha thalassemia and beta thalassemia. If there is a defect in alpha chain manufacturing, then alpha thalassemia is present. It is generally due to the gene deletion or mutation. And the second is beta thalassemia, in which the beta chain is not produced enzyme. The clinical features of thalassemia are microcystic hyperchromic anemia, which can be observed in clinical findings. Also, in lab report of blood samples, target cells are observed. Target cells are circular cells with a dot in between that looks like a bullseye. And crew-cut skull can also be viewed on X-ray of a thalassemic person. When a person has thalassemia, hair-like bulges are seen on his X-ray. Now, the alpha thalassemia is further classified into 4 types. In normal, the 4 genes that are present on chromosome 16 are normal. all are functioning efficiently. But in affected individuals, if one gene is missing, then it is silent carrier type of thalassemia, in which no symptoms are visible due to all three genes performing efficiently. If two genes are missing, then it is known as alpha thalassemia trait, and minor anemia can be observed. If three genes are absent, then it is known as hemoglobin H disease, and it presents with mild to moderate anemia, but a person can perform daily tasks efficiently. If all four genes are absent, then the condition is known as hydrops fetalis, and the fetus will die as soon as it is born. Likewise, the beta thalassemia is also of two types, in which the gene present on chromosome 11 is affected. If one gene is absent, then it is known as thalassemia minor, or if both of them are absent, it is known as thalassemia major. Thalassemia major is far more severe than thalassemia minor. Now let's understand the role of genetics in inheritance patterns. As we know, the thalassemia is autosomal recessive disease. If the affected gene are carried forward to the children, it shows 25 probability of both the son or daughter being affected. If the unaffected genes are transferred in their children, 25% probability says both of them will have disease, or else maximum percentage says that they will be carried if only one affected gene is carried forward to their children. The diagnostic tools will be explained by Nishish.
Diagnose myself, Rishikesh Patel. I will continue the diagnosis. First, complete blood count. A complete blood count helps us to detect anemia by measuring hemoglobin level, red blood cell count, and their size. Another one is taking a blood smear. Here we can distinctly see abnormally sickle cell cells. The next one is hemoglobin electrophoresis. It based on separation and charge and size. It helps us to detect the different abnormal forms of hemoglobin acid. Some labs may have high performance in liquid chromatography. It helps precise quality of hemoglobin is present. The treatment will be discussed by my friend.
Treatment of abnormal hemoglobin. So treatment of abnormal hemoglobin depends on the specific type and severity of the condition. There are some specific approaches. So medical treatments. First one is your blood transfusion. So blood transfusion. Regular blood transfusion can help increase oxygen delivery when you have no budget for the bone marrow transplant. Then you can use this method, that is blood transfusion. Then second one is the chelation therapy. Chelation therapy used which removes the excess iron from the body, often caused by the frequent blood transfusion. Then third one is the medication. So various medications can help manage the symptoms, such as the pain medications for sickle cell anemia. Specific treatments. Then sickle cell disease, hydroxyurea, pain management, and blood transfusion. Then thalassemia. In case of thalassemia, blood transfusion and chelation therapy and potentially bone marrow transplantation are the methods used for the treatment of thalassemia. Then emerging treatments, gene therapy. Researchers are exploring the gene therapy to correct the genetic defects. Stem cell transplantation. Bone marrow transplantation can potentially cure certain hemoglobin disorders like sickle cell anemia. Then lifestyle changes, stay hydrated, drink enough water, plenty of water, then manage stress. Find ways to manage the stress and avoid extreme temperature, that is avoid extreme heat or cold. Thank you.
So let us have a quick summary of it. So in this case, we understand about the normal hemoglobin, the abnormal hemoglobin structures, what are the point mutation changes or the structural changes. Then we understand from the state about the sickle cell anemia, and then we understand about in detail about the sickle cell anemia from our batchmates, and after that we have seen these thalassemia cases also, and in that we understand the preventions, treatments and diagnostic tools for the abnormal hemoglobin. So here we all conclude our seminar. Thank you.
Okay, any questions? खाली उभं राहा नाही तर वरती उभं राहा.